Hello, everybody. Thank you very much for listening in today. Welcome to CrownCast. This is the new podcast from Crown Bioscience that aims to discuss the emerging trends, breakthroughs, and viewpoints in oncology drug discovery and development. I am the host for today's podcast, and it's my honor to bring you conversations with scientific experts from around the globe. My name is Jonny McMichael. I'm Crown's VP of Client Experience and Enablement. I'm based in Edinburgh in Scotland, having spent the last 10 years with Crown Bioscience. I do apologise for my accent if you're not used to the Scottish accent. I'm not making my accent up to sound a bit like William Wallace or Mel Gibson. So hopefully bear with us throughout the accent challenges. Today's episode, I'm delighted to be joined by my good colleague, Enrico Pesenti, who is Crown's Executive Director of Client Engagement based in Milano, in Italy. Enrico has been with Crown for the last 2.5 years, working in leading our scientific engagement and consultancy groups. Prior to joining Crown, Enrico's had a long and distinguished career in the drug development industry. Spent a number of years in leading scientific roles with pharma companies, including Pharmacia, Nerviano, and also spent 10 years as the CEO of Accelera, a regulatory preclinical CRO in Italy. And today's podcast, our topic is drug resistance, a topic very close to Enrico's heart and scope of expertise. So, Enrico, welcome to CrownCast. It's a pleasure to have you on today. How are you? How is Milan?
Yeah, bit cold, but it's nice, and we are waiting for Christmas.
I doubt it's as cold as Scotland, Enrico. So enjoy the sunshine. Enrico, thanks for coming on. Really appreciate your time. And I guess as a start, would you perhaps like to just give us a little bit of background to yourself and how you ended up where you are at Crown Bioscience?
Yeah, sure. You can see from my hair that I'm gray, so I'm on this job since a long time, and I started very young. I was always working in oncology research and mainly in pharmacology. So, as you mentioned, I spent many time of my career in working in pharma, in drug development. I had, despite being old, I was lucky to see the start of what was molecular oncology and new therapy with target therapy. We had a very strong program of kinase inhibitors, and two of these are actually at the moment at the market. So also some tangible results of what we have done. Then along the career in big pharma, then as you said, I spent almost 10 years as CEO of Accelera. It's a preclinical CRO, so I started to get experience in the CRO worlds and to see drug development from the other side. And then after this, I end up in Crown, trying to bring my experience and my knowledge, especially in pharmacology, helping Crown and our clients. And yeah, today the topic is a resistance. I think is something to say is in some way the dark side of our work. And in one side, I'm still amazed when I see and I learn more and more the complexity of cancer, and I still see that continuous progress we are making in understanding this, especially with the new technology. We understand much more of molecular pathogenesis. And what I see and make me really in a way happy is that what we were always thinking about and making personalized medicine is becoming true. It's actually there. We are doing personalized cancer medicine. And this because in the last 20 years, we have seen the development of different cancer therapy modality, including small molecule inhibitors, kinase inhibitors I mentioned before, but up to monoclonal antibody and checkpoint inhibitors, cell-based therapy. I think in the last 10 years, there were something more than 200, 300 new drugs approved by FDA. And I think 1/3 of these are new mechanisms of action. And all these drugs now are part of the standard care alone or most frequently in combination. So that's really, there's a really impact. And this is something that makes us, I can say, proud of our work and of our small pieces of what we are doing, each of us contributing in a different way. And you see the impact on patients is there. I was reading yesterday the numbers of the decreasing of the death rate in some cancer in the United States, breast cancer, lung cancer. Of course, it's not only treatment, it's prevention, early detection, changing our lifestyles, but new therapy are having an impact. And I'm thinking, when I started, there was basically only chemotherapy and lung tumor. The only therapy was cisplatin. Now you think about non-small cell lung cancer, we have, I don't know, tens of drugs, each drug specific for each specific mutation, for each specific patient of different therapy line. Another example is melanoma. There was only the dacarbazine, basically not active. Now we have immunotherapy, specific small molecules. And I think this year was the tumor infiltrating lymphocyte-based therapy for melanoma. So it's a big advance.
Yes, yeah, so we're certainly a long way off what was once the one-size-fits-all approach to cancer treatment, where a patient would come with a...
Yeah, it's true. But as I say, but as I said before, there is also the dark side that most of these patients, especially the one that they enter treatment with advanced disease or metastatic disease, they experience resistance, and that is manifested as local or distant relapse. So cancer is relapsing, and relapsing after basically all the available therapeutic modalities is common. And this is because every therapeutic modalities leave behind some cells that are not affected. So what is called minimal residual disease, that is, it's basically a reservoir from which relapse emerges. So it's clearly that the ability of malignant cells to develop resistance to initially effective therapy is the major obstacle that we see now in cancer treatment, and definitely with a big impact in patient outcomes, and is also one of the biggest challenges we have in drug development. That's the general picture where resistance is and the value of this small word.
Got you, exactly. And so, yeah, really to kick off, you've touched on it already a little, but would you be able to talk a little bit about what you see as the main current challenges in oncology drug development?
Oh yeah, let's start with the definition. Being scientists like definition, because make the word the complex, giving some order to the complexity. So drug resistance is the ability of cancer cell to survive and proliferate despite the treatment you are giving. Treatment that are meant to kill or to grow, to inhibit that growth. And this is done through a very broad spectrum of mechanisms, and you will see later on, maybe we can discuss about this. But in generally, the definition is that resistance can be primary or intrinsic or secondary or acquired. Primary means that basically there is no objective response to the treatment following therapy. And the secondary, you have response, but then you have relapse. So this is clearly a classification based on the medical aspect. If you change your point of view from a biological point of view, where it's much more complex, also in this case, despite the several mechanisms behind resistance can be categorized into major groups. The one where therapeutic resistance is a result of a genetic evolution, a selection of genetic mutation that is enabling cells to escape therapy. So basically, there is one cell or a group of malignant cells that they have already or they acquire specific genetic mutation that give the cancer cell the capability to escape treatment. It's a kind under therapeutic pressure. It's a kind of Darwin concept: pressure, selection under pressure. and only the few clones that already have, many times they already have, is very similar to the resistance to antibiotics, whatever. But what is very interesting and open up some different point of view is emerging that there is another kind of resistance that is not genetically based, is what is called is an adaptive mechanism. So we have cells, a subset of cells that basically under therapeutic pressure, they enter a status of, let's say, drug tolerant. They are called persister cells. And this is done by post-transcriptional reprogramming. And a couple of examples, chemotherapy is very well-known phenomenon. It's called epithelial to mesenchymal transition cell from different cancer. Epithelial cancer can change their phenotype, acquiring a different phenotype. It's more indifferentiated, like mesenchymal, and this is connected to chemoresistance. It is pretty interesting in melanoma, where these two status, these two phenotype can basically switch back and forth by the cells depending on the pressure of the treatment. And one of these phenotype, what is called invasive, the mesenchymal one, is resistant to MAPK treatment. So it's giving specific resistance. And under pressure, you see most of the cells are transitioning to this status. So under the pressure, under the stress of the treatment, cells are changing, assuming a status that is basically not responding. That is very interesting. And behind, basically, it's like the tumor cell is hijacking many of normal mechanisms of survival of the cells and transforming these in a mechanism of resistance. And this is there is activation of different pathway, or as also for immunotherapy, you see activation of different checkpoints and as response to anti-PD-1 therapy. So it's a very complex and sometimes intrinsic of the basic mechanism of cells. And what is emerging is interesting, is a bit a contradiction of what is our theory of Darwin therapy, is that at the beginnings, these adaptation phenomena are reversible, but under therapeutic pressure, they can be irritable. So cells stay in this status and they can proliferate maintaining this status. So that is a kind of it's pretty interesting, but it's open up also new avenues of therapy, of course. And what I'm thinking, talking about drug development, I would say that dark side is a kind of yin and yang. You have new drugs, but you have also tumor cell adapting to the new drug. So you get one, and then you get the response of the cell. So it's something, and it's well known from the early days. I was not born yet, but at the beginning, even with the first use of radiation, I think at the beginning of 1900, there were cells, not tumor normal responding, but the real emerging of resistance was with the introduction of chemotherapy. After the Second World War, we have the first patient treated with chemotherapy, more or less. And it was pretty clear that using methotrexate, cyclophosphamide, or the new chemotherapy at the time, there was clearly initial response, but then relapse, not in most patients. So the next step was to use combination therapy. So putting together different mechanisms of action. And this was very effective for leukemias, lymphomas, or whatever. But also in this case, there was relapse. But until, I can say, the early 80s, it was not very clear the mechanism. And one of the first mechanisms, I remember very well, because when I started working in the lab, was my lab focused on the multi-drug resistance. In the group, there was basically the doxorubicin was discovered in Nerviano, where I was working, and so there was a long tradition of evolving the chemotherapy and resistance was the problem. So we started to understand the mechanism behind the mechanism. So MDR mainly was due to P glycoprotein efflux. So basically, cells is a defense mechanism of cells from external chemicals. So you pump immediately out, and you decrease the intracellular concentration of the drug. So you become resistant. But then with target therapy, we saw that we understood much better what is the genetic base that induced resistance, that is kinase inhibitor, all the point mutation in kinase structure that are given to the kinase domain. Basically, the inhibitors are no more active for different point mutation that can keep the protein, the kinase domain in active status, or they can block the access to the ITP binding pocket. So different mechanisms, all giving specific resistance. And I record making a lot of in vitro model, bearing all the different mutation and testing hundreds of compounds, and a lot of interaction between cell biology and medchem at the moment. And what interesting is that this kind of study, they brought also changed also some dogma. No? Chemists, they were avoiding completely any covalent binders at the time, but then they discovered that maybe some covalent binding on the kinase domain was a way to overcome resistance. Osimertinib is one of these, for example. So it's interesting how you learn in the lab the things, and things are evolving. And then, of course, now we have, as I mentioned before, immunotherapy, other breakthrough. Each of new therapies bring a new problem, so a new mechanism, and this is the base. I can say also in this case, the complexity is a challenge, but it's bringing also opportunity. So understand the mechanism, open up therapeutic approach, and we can discuss about that. Like, if you want, later we can go.
Yeah, absolutely. That's fascinating. I've actually learned quite a lot in those 10 minutes, Enrico, about drug resistance. I mean, of course, resistance means different things depending on which area of biology or drug development you work in. My first experience when I worked in industry was not drug resistance. I was a medical microbiologist, and my focus was on cystic fibrosis, and there the main problems were multi-drug resistant microorganisms, bacteria that pharmaceutical companies back in that day, the likes of Pathogenesis Corporation, were working on subverting that resistance and getting around it to really try and improve patient outcomes, because these are very that level of resistance is very debilitating and really prevents your ability to treat the condition. So that kind of leads me to ask you. You've talked a little bit there about the impact of drug resistance on the development of new drugs and what it means for drug development approaches. What would you comment on the main challenges we see in the clinic related to resistance? What are the patient impacts?
Oh, that's the real question, because at the end we are talking about patient always, and and in clinics the major problem is pretty obvious. You have the outcome, the clinical outcome is impaired. So if you see the numbers of in tumors, in breast tumor, for example, most of the patients responding to target therapies become resistant in relatively 1-2 years. The same happened to as soon in tumor like lung, as soon as resistance emerge, that immediately the performance status of patient degraded. So the outcome is really poor for patient. And one example, if you take pediatric tumor, I think there is neuroblastoma that is a rare tumors, but relatively frequent in pediatric tumors. And I think that 50% of patients, they have relapse, even if they are responding pretty well and are complete remission. And these patients are patients that are going through radiotherapy, complete ablation of bone marrow, of chemotherapy, immunotherapy. They go all through different line of therapy, but then become relaxed. And this is telling also how cancer cell can adapt. Also in this case, to different point and different way of treatment. That is really, I would say, not amazing, but is really impressive. And then, of course, there is an effect of not only to the disease outcome, also on patients. If you think that the impact on the psychology of the patient, so it's not only a matter of medical or biological problem; it's a real problem for the psychology of the patient that become immediately no more responsive. But also from a medical point of view, you run out of options. And even in clinical research, when you get resistant within the clinical trials, then the responses become very difficult to interpret, also the biomarker. So it's really really affecting in a profound way all the outcome of the clinic. And this is, again, why it's so important to find the right strategy, both in clinics, but also in discovery. What we are doing that in early research, to find the right strategy to overcome resistance.
Yeah, perfect, Enrico. That's fascinating, and I really enjoy hearing your insights around the real impact to patients, because we get so often very focused on the drug development aspects and forget the ultimate aim is to make the lives of patients easier and more manageable. So in that regard, you mentioned strategies. Are there any new strategies being developed to overcome drug resistance that you could touch upon?
Yep, there are many, and say before, there are many opportunity both in the bench and the bed, and vice versa, because from the clinical experience we are learning a lot, and dissecting the patient response or non-response, we can learn more and more and more. And this is basically due to the new technology we have now. What I like very much, for example, now, one of the approaches is using precision medicine and a biomarker-driven approach. And now this is possible mainly. What a technology I like very much is the single-cell multi-omics technology. So it is the response of what is one of the major problems I was not mentioning, that is tumor heterogeneity. If you think about the different resistant mechanisms within the same cells, if you multiply this for the million of cells per tumor, you get immediately the idea of how heterogeneous cells with different cell population, with different grade of adaptation, with different grade of different mechanisms. That I forgot to say that all these mechanisms, sometimes they are happening at the same time, and not only in the cells, but also the interaction of what is around the cells, that is the tumor microenvironment. So it's a really dynamic situation, dynamic and plastic situation. So tumor heterogeneity is really one of the major others, the challenge that we have in treated tumors, understanding the tumor biology. But for example, I take the example of single cells multi-omic technology, or single cells or single nuclei multi-omic technology. That is really revealing, make us to understand much better the tumor heterogeneity, pointing out the different tumor population, or identify rare resistant cell population. That is very important for resistance, where maybe the mutation or the adaptation of treatment occurs in a very, very few cells. But with this kind of technology, you can identify this and have a map of also the different status. for example, as I say before, the transition between the epithelial and mesenchymal status can occur at different stage, at different time of different cells. And with this kind of analysis, you can identify the different stages. I was mentioning before the neuroblastoma. There is a very, very recent, published this month, paper from the John Mary's group of the Children's Hospital in Philadelphia. They were analyzing, I think, more than 20 cases of neuroblastoma with single nucleotide technology. They identified, I think, at least 4 different subpopulation of cells with different phenotype as a result of the treatments. And between these two, they identify some specific pathway, for example, MYC inactivation or nucleofactor kappa B activation pathway that are typical of resistance. So having the possibility to do this kind of very in-depth analysis, they were analyzing, I think, more than 200,000 cells, is giving you insight of the mechanism and open up therapeutic possibility. Even if, for example, on NF-kappa B, we know that is a pathway that's very difficult to touch because of is an essential intrinsic cell pathway in the cells, so can lead to toxicity and so. But you can then look to downstream pathway of the activation, beta. So it's open up new horizon of treatment in this case. And connected to this, what I like very much is now we are adding the third and the fourth dimension of the tumors with spatial transcriptomics. So we see the 3D, but also we have a possibility to insert also the timing. So there is the possibility to follow evolution during the time. So it's becoming more and more detailed. Plus the typical multiplex immunofluorescence that you can see of the protein markers or whatever, especially very important within cell-cell interaction between tumor cells, immune cells, and tumor microenvironment. So that's what I like very much in terms of analysis technology applied to biomarker analysis of clinical results. But from our side, I can say for experimental models, of course, I think having PDX or patient-derived xenograft, or in generally patient-derived tissues of sample is a real advance. PDX in vivo, everybody knows, especially in Crown, we know this. It's our strong points. And then in vitro, we have the organoids and all the other culture system using patient-derived tissues. And it's giving, for example, if you take organoids, the possibility to study also the interaction with the tumor microenvironment and the other cell, not only immune cells, but also fibroblasts, tumor-associated fibroblasts. So, and using co-culture technology. And sorry, and you have also a kind of throughput screening. You can discuss if a high throughput, medium, or low throughput screening in vitro to test, for example, combination and personalized combination to the patient. That's the real advance. And I go on. So other technology I like very much is all the genomic editing possibility with CRISPR. So you can, one example, you can create some specific model or with specific mutation, and we have some example of mice bearing different mutation of KRAS mutation. But one of the problems coming is the massive amount of data that we are seeing. And so at this point, is where computational and AI-driven approaches are entering. So one side, now I think every podcast at some point are mentioning AI, but I think it is very important. And what we are machine learning and calculation power is very important to analyze and making deep analysis of the massive amount of data that we have. And there is one example I like very much is you can also actually, for example, in the case of kinase inhibitor, you can actually predict the weak point, the most probable point of mutation conferring resistance. And then you can do this in the lab, not in the clinic, after some years. So that is, you can understand how this can, which kind of impact can have this, because you bring immediately to the patient a drug, or you know already what is going to happen, so you have not to wait 5-6 years to study the next resistant mutation that you get. And it was applied, for example, for a track inhibitor, track inhibitor was used, and we were really predicting potential mutation given the resistance. That's one of the few examples. And then last things is all the thing related to digital pathology that can really, say, go much more in detail, identify patterns that maybe we cannot do with the simple microscope. So it's incredible. I think it's really exciting what's going on. But sorry, the last point, and then in the clinic, in the practice, what is the result of everything, or what we say is the combination. So we have tens, hundreds of drugs we are learning to use in the best way and in a personalized way. So now we have biomarker that can drive the personalization of the medicine, and we have also research tools like in vitro, as I mentioned, organoids, or an algorithm to analyze to make combination much more effective and personalized to the specific resistance pattern that are for the patient. You tend to see the general picture. I like very much when you see all the pieces coming together. No? And then you get a bit with the experience, and also some observation that you have done maybe 20 years ago that are coming back and with a different point of view.
There are a number of themes there as drug development has evolved over the decades, that there will be themes that will, come back. There will be models that will come back into front. Yeah, exactly. I mean, I guess I've really only got one more question for you from what you've said. So you talked about the important the importance of approaches to model or try and develop strategies to predict and also deal with the implications of resistance. You mentioned things like modeling the TME is important. Yeah. The more translational the model, the better, using AI as an approach to, again, direct that modeling. So as a CRO, we have a lot of companies come to us who are not experienced in certain areas, or with less experience, or moving into an area for the first time. So what advice would you give to a company coming to us, telling us that resistance is a concern for them, or they're wishing to target an indication or a therapeutic modality for which resistance is a challenge?
That's a good question, Jonny. The first thing is that I learned is that even if several of my bosses were already asking about this, that the clear experiment doesn't exist. It's a complex work, and it's never black and white. It's never yes or no. But what is important is to have in mind, try to keep in mind your general picture, your goals at the end, where you want to go. And as you mentioned, I want to explore and dissect in a specific clinical situation, a specific indication. I want to position my new drug in a specific setting, could be, I don't know, second line, third line. So having a strategy helps, and the general picture. And then based on this, you choose the right path going there. So the right experiments and the right path. And being a CRO, what we need to give is to put on the table all the possibility. So we need to have all the technology, but at the same time, we need to have the capacity together to find the right way to use it and the right path. And because at the end, what we want, first, yeah, we need to create a win-win situation. The success of the drug, of the project is also our success, but definitely, ultimately, it's the success of the patient that will get this job.
That's been fascinating. As I say, I will watch this podcast back myself to learn more myself about drug resistance, because I've learned a lot there already. So do you have any closing remarks or comments on drug resistance?
Yes, more in general than drug resistance. Last month we had a roadshow in Switzerland. We were making some presentation, meeting clients and collaborators, and I had a couple of slides showing all our models, especially model focus on resistance. So it means sample coming from patients where we know the story of the patient, we know the different therapeutic that they got. Some of them are longitudinal samples, so we can see the evolution during the time. But then I was really looking at each single patient that, of course, for us are numbers, but behind there is a patient, there is some person. And you see that what this patient had to do. And so some of them, they had 8 lines of therapy. And anytime we new therapy, because there is response, relapse, new therapy, then I thought, so it's some way touching, because it's when you made the translation, you mentioned at the beginning from the lab to there is a person behind, and at the same time give you the strength,
The
the motivation to go forward. Yeah, that's this something was touching me. So now when I'm seeing these slides with a long table, I'm always thinking about this.
Absolutely, exactly. We have to bear in mind the patient is waiting, and that's the important thing. Here's to hoping that all of these novel approaches and all the ways in which we're able to model resistance in the preclinical and clinical phases will allow us to find new approaches to subvert resistance and find those therapeutics that will make a major difference to people's lives. So thank you very much, Enrico. That's it's been a pleasure speaking with you as always. I could sit and listen to you all day. My wife won't let me do that. So thank you very much for your time.
My wife too.
Really appreciate it. So thank you. And thank you for everyone listening into this this podcast. As we set out on CrownCast, is a fairly new venture for Crown. And if you've listened into this, we really greatly appreciate you supporting us in this, and hope that you've got something out and enjoyed it. If you have enjoyed today's episode, please do be sure to hit subscribe on Spotify or on YouTube. And you can also find CrownCast directly on crownbio.com. And if you have enjoyed it, please do join us for the next episode of CrownCast, as we'll have pretty varied topics on here and guests talking on pretty diverse topics. So thank you very much. Grazie mille. Take care.
Bye.